Okay, again, as a review, what we are going to do is we are going to download the gene list that you created into this program, Ingenuity Pathway Analysis. What this program does is it'll interrogate our gene list for all these overrepresented biological processes in it. Again, this is probably the most powerful bioinformatics software on the planet. I would take this program over a whole room full of postdocs any day. What's a postdoc? These are like very cheap labor for science. So these are people that have gotten their PhDs and they're looking for a position, and you can hire them for very, very cheap. Yeah. Going the academic route in science, it does not pay well. I'm just going to let you know right now. So don't ask me to borrow any money. Okay. Is everybody on this page? Everybody has their Excel spreadsheet downloaded. Okay. This will be, so what we're going to do is we are going to take those genes that were different in the grade 3 lung or breast cancer, because that's when the survival rate drops dramatically. We wanted to see what genes were important in that stage. Salud. So what we're going to do is we're going to download that list and then basically use this program to see if we can learn anything about stage 3 lung cancer. So let's do that right now. And then for the rest of the period, I'm going to let you work on this. So does everybody see this new button here? Yeah. Click New Core Analysis. Rod, are you with me? Okay. If you cannot see, you might want to move closer to the screen. Yeah, so we're going to go upload a new data set. This window, this Create Core Analysis, is going to upload a new data set. Here's what you're going to do is you got to find wherever you put that thing. I put mine in my Dropbox. GBR. There it is. And breast cancer. All right. Does everybody, once you've done that, does it say on this raw data, does it say 971? Yeah. Yay. All right. That's perfect. Now what we got to do is we have to tell this software what our IDs are. What column gives us how is this program going to be able to determine what we put into it? Given in this first column is the gene symbol. So this is the symbol that correlates to any, the genes that we looked at. Again, you can look here, this top one, depth domain count, whatever, was one of my top genes. It was in 87% of my studies. And it goes up. So here's what we're going to do is we're going to select this first column. We're going to say ID. That's where my genes are. That's where this program is going to learn where my genes are. And I'm going to tell you number 8 is Aurora kinase A. What's that? ARCA. AURKA. If you look over here, this is the gene name. You can see Aurora kinase A. This is that gene that Dr. Torquia. Did I say? Wait, no. Torquia. Torquia. Dr. Torquia basically is working on. A lot of these genes and what you're going to see are important in other kinds of cancers. And that's what's going to help you find the targets that you want to present on at the very end of school, is finding something that you can describe that kind of, describes this stage of breast cancer. Okay, so ID, here's what I want you to do is go over 1, 2, 3, 4 columns and put observation 1. Are you with me? Oh my, quit smelling yourself. That can't be a pleasant experience. All right, observation 1, here's what I want you to do. Percent studies, I want to include this information. I'm going to put other, experimental other, expression other. Yeah, just put observation 1. You want observation 1, and then under the window right underneath it, you can have all these different choices. Pick that EXP other. Okay, here's where it's important. Rod, are you with me? I'm going to check to make sure that your gene list is downloaded. If it is not, you've got a problem. Well, then talk about. Maybe if you're not talking with G Money over there. Yeah, is it almost summer yet? I think so, right? Summer break. That's what I meant. Okay, so in the next column, you're going to put observation 1. Here we're going to go EXP fold change. This is how many times that gene went up or down in the stage 3 breast cancer compared to the other stages of cancer. That's the amount of times that gene went up or down. So it's basically producing twice as much mRNA at grade 3 breast cancer than it would be at, say, grade 1 breast cancer. So this gene is much higher in the more advanced stage of breast cancer.
The ones with negative for tumor suppressors?
It may be. Way to think about that. Yes, JC, that's probably right, actually. But we'll see. It all depends on who they interact with. Again, remember, these are our potential criminals and cops. And this is we're going to use this information to figure out, what kind of crime was committed and how they did it. Okay, everybody's got these selected, right? Hit save and create analysis. And you'll get this thing 1 or more fold changes. Don't worry about it. Hit OK. Okay. You can save this as the same thing. I got to save mine. I'm going to save it as 2 because I already put this in. But you can just leave the name. Here's what I want you to do, though, is choose a new project. Make this breast cancer. So under that create project, put a name breast cancer. If you haven't done so already. I already have one. You got it?
I saved it.
You saved it? You already got breast cancer? That's good.
Yeah, I did a new project though.
What's that?
I did a new project thing. I just click save and then do this new project stuff.
Yeah, I think it went into your first one. So oh, wait. Okay, you're fine. That's all right. That's okay. It'll be in your first folder. You'll see it. And if not, we'll fix it. Okay, hit save. If you've created a new folder. Okay, this window will come up. This is basically the program's asking you, how do you want to analyze these genes? And there's all kinds of options here. You can just look at the things that go up, things that go down. You can do all kinds of stuff. Here's what I want you to do, though, is we're going to leave everything just like it is and hit run analysis. Again, the same name could come up. You can just use the same one. Hit OK. What we have just done is we have taken our list of, how many do we put in here? It was like 900 and some, right? We took our list of 900 and some genes. We sent this to a supercomputer in Silicon Valley where they are using their database to tie all this biological information to our genes, and then they're sending us back the results. And that's why it takes a little while. In the meantime, though, I just kind of want to show you, this is what I want you to be looking at, is start pulling out some of these relationships that you can look these genes up by yourself. That if I look, one of my top, where's my top gene? Wait a minute. Come on. Takes a while. There it is. This last page gives you all the molecules. I can sort this by the p-values. Well, actually, no. Experimental. This abnormal spindle thing was in my top list. It was basically, it's got a high fold change, and it changes in all these studies that use that stage 3 breast cancer. Here's what I want you to do, and this is how I want you to kind of play with this. I'm like, oh, okay, this must be important breast cancer. I wonder what it does. I can put this ASPM, and just go to a new tab in Google. I can go ASPM gene, and then put breast cancer and see what pops up. Yes, you can totally use NextBio. In fact, that might be a great place to go. So I just looked up here, dysregulation of this expression are correlated with epithelial ovarian cancer progression. And this will tell you probably why it's probably involved in this cancer stuff. This would be information that if you were going to present on this gene, you would include some of this. Hey, I found this gene. It's in all these studies that we looked at where they have the stage 3 breast cancer. And by the way, it's also found in all these other cancers. That would be information you want to present. Then you would basically come back here and you could say, figure out what is this involved in. It's in network 1. I can click on this. Oh, wow. Look at that. Which one's the Godfather, guys? Yeah, totally. That might be a good gene. I've been looking at this gene. It goes down. We measure it going down, but everything that I read on here says it's like more active. So something's broke there. Like this is very interesting. But yeah, I mean, that's what you look is, look at some of these networks, kind of look and see what are the genes that are like connected to everything. Look them up, see if they make sense. Obviously, this gene goes to this one, which is, I bet this is a huge connection here. And you could talk about that. How does this affect this? This is normally supposed to induce it. What's going on here? All right, so for the rest of the year, I'm going to let you work on this. Our last day of school, which is May 2nd, I think. Last day of class, is that right? May 2nd? May 2nd, we will present. I'll try to bring in something special for that day. So don't get kicked out before then, Rod. G-Money. Okay.